All right, welcome everyone to the N of One Collaborative Seminar Series. My name is Nicole Nolan and I'm N1C's Programs Manager. It's our pleasure to have you attend the first seminar of 2026 with wonderful speakers. First and foremost, we want to thank our partners, the Chan Zuckerberg Initiative, for their support of N1C's mission. I'm going to drop my email in the chat. If you or your organization would like to get involved with N1C and learn how you can support the mission, I would love to hear from you. Oh, hello, Tim. It's good to see you too. One last housekeeping bit before I turn it over to Winston and Julia and Tim. Winston, who will be moderating today's session. We would like to invite you to Connections in 1C's virtual collaborative platform following today's call. This meeting is being recorded and will be hosted on Connection, and we will be continuing the conversation. So we look forward to seeing you all there. All right, without further ado, Winston, N1C's co-founder and president, please take it away.
So I am moderating this, but we happen to be in DC together for a really exciting announcement today. So I'm actually going to pause and turn it over to two very important people in this entire field and have them share a little bit about what happened today, as well as how it relates to the seminar that you're about to hear. So, Julia.
Thank you. Thanks, Winston. So today was a very, very exciting morning here in Washington, D.C. We just came back from HHS FDA announcement of this new plausible mechanism pathway, which basically is the many, many, many years working up to being able to see individualized medicines not being done one at a time for one dying child like my daughter Mila at a time, but really kind of scaling that to the masses. And so Tim and Winston and I and a bunch of other parents and doctors were all part of this announcement. And I think what it means is it means that the FDA is willing to do things differently. They're willing to, instead of see one medicine for one disease or one child at a time, is to be able to group them together and lead up to what should look more like an approval of the way we make medicines and less about, the actual medicine one at a time. And it's very timely for this discussion today with Michelle Mellion and Paul Gisson, because in the UK, in parallel, the MHRA has given the green light for a very exciting master protocol, which is very similar to this. And I think it just sort of lends to the direction we're going. And so hearing about how Michelle at Everyone Medicines and Paul at Great Ormond Street Hospital work together as a company and an academic hospital together is just a great example, which you'll hear about today, of a model of like how we might begin to think about doing this at scale in the UK and in the US now. So this is just very timely and very exciting. And I don't know, Tim, if you wanted to add anything to that, but
I'll just say then we have homework after today's seminar, which is after you finish hearing from Paul and Michelle, um, pick up your local laptop and hop over to the HHS website and see how what Michelle and Paul are describing could be applied under the new guidance. I think that that's the charge that's been put in front of us today by HHS. With that, I'll just, yeah, not take any more time and give it back to our presenter.
So this really was unplanned, right? We found out about this and we are now, I mean, you can probably see that it's unplanned. We're sitting here with a laptop and a stack of books and well, Winston, Julia, Tim name tag. But what I want to share with you is that this is the purpose why the N1C exists, right? We're really trying to share with you the cutting edge of what's possible to operationalize and create the field of interventional genetics, genetic surgery. This is the next steps of taking guidance and actually turning it into treatments. And so we are here right here with you. And what I'm going to do right now is turn it to Michelle and Paul to introduce themselves. And what the format of this is that there will be a talk that Michelle and Paul will share with you. We will go through it. And have a chance to answer questions in it'll be about 40 minutes of talks, but you can drop your questions in the chat. And then the last period of time will be discussions. And as Nicole said, please follow up on the N1C connections. This is a place to continue the conversations far beyond just the hour of the seminar. So.
one last note would just be is this is a very international group, is that this is not about how amazing work in the UK is happening or about amazing news today in the US. But the idea is that this is the beginning of many other regulators in many other countries beginning to sort of imagine individualized medicines, including what you'll hear about in this talk, how this becomes scalable and sustainable around the world. So my hope is that all of you will engage us as needed or anyone else and really try to push your regulators to look at the MHRA and the FDA, and everyone begins sort of thinking about this in a very different way. So this is a global problem. It's a global, that's an understatement. It's a global health crisis with hundreds of millions of patients with genetic diseases, and now the ability to potentially have highly effective individualized medicines. And it starts with the regulator in many cases. So I think, Michelle, pass it off to you.
To you, Michelle. Thank you.
Wonderful. So thank you, Winston, Julia, and Tim. Congratulations to you all and to the N1C. This is really the beginning of a new era, I think, as we move forward. And so I'm Michelle Mellion. I'm the Chief Medical Officer here at Everyone Medicines, and I'm joined today by Professor Paul Gisson from Great Ormond Street Hospital, who is our lead investigator. And today we are going to be talking about how we've partnered together to build a master protocol to advance development of individualized medicines at scale. And so here are disclosures. And many of you may have already read the abstract, but the session today is going to describe a collaborative model in which Everyone Medicines and GOSH have developed this master protocol to advance the development of individualized medicines beyond one-off compassionate use. And as Julia had alluded to, is that this protocol is already approved by the MHRA, who has been a very forward-looking regulator in this space. And we have already enrolled and dosed our first patient in this protocol. And the first patient that has been enrolled under this protocol really represents a critical milestone for our community because it demonstrates the operational feasibility of our approach. So this webinar will introduce the master protocol, which is a prospective clinical framework that is designed to enable standardized data collection and pre-specified analyses across individualized ASO therapies for patients living with unique genetic variants. And really, this is the first part in generating the data that's needed to support a process approval. I know that you are all very aware of this in this community, that, we have had some huge scientific success. We can now diagnose ultra rare genetic diseases rapidly, but for the most part, it's difficult up until hopefully now to provide timely treatments. And we have all been living through this extraordinary era in rare disease where we can sequence a genome in days, identify pathogenic variants in weeks, and we can diagnose conditions that 20 years ago would have remained invisible. But the brutal truth is that follows this scientific triumph is that as many families go on this diagnostic odyssey, it doesn't end with treatment currently. What it ends with is really abandonment. And because the moment the family receives this diagnosis, they discover something else. They discover that their child's mutation is too rare to matter in the current system as it has been developed, in this development system. And when that happens, it doesn't matter that we have the technology, that we have all the science for these treatments. Our system is just not built to respond to these people living with ultra rare genetic variants. And so this isn't not necessarily a specific rare disease problem. This is a problem that really affects our patients living with ultra rare genetic variants. And because if only one child or a few children in the world have a particular genetic variant, the standard model that we are currently living under essentially collapses, right? There's no trial feasibility, there's no commercial incentive, there's not an obvious regulatory pathway, there's no evidence generation strategy. And so what we've been doing up until now is, families have been fundraising, academic labs have been improvising, clinicians have been working with their patients to get access to much needed therapies. And this is all heroic improvisation. But as we all know, in practice of medicine, that medicine is a system. And so the tragedy really here is that the science that we have today is ahead of the infrastructure. And all of this had started to change actually in 2019. And what that change was Millicin. And as we all know, Millicin is a bespoke antisense oligonucleotide that was designed for Mila living with Batten's disease. And at the time, it's really an astonishing demonstration of what is scientifically possible. And from that came many other therapies, including the most recent success with baby KJ. But all of these therapies and even the success with baby KJ have not solved our individualized medicine problem, getting these therapies to people that need them most. And what we have shown is that we've proved that it's possible, but this being possible is not the same as being repeatable. And medicine, as we all know, because we're scientists and we live in this framework, medicine is defined as not what can be done once, but what can be repeated, what can be regulated, what it can be standardized and delivered without relying on a miracle. And so in 2022, Giulio Vitarello, Mila's mom, and Dr. Tim Yu from Boston Children's co-founded Everyone's Medicines. And it was really inspired by their journey to create Millicin. And their hope really in creating Everyone Medicines was to remove the financial and scientific burden from patients and their families and make these therapies available from all who can benefit. And so that children, just like Mila, can have their lives improved by these medicines. And we know compassionate use is necessary, but it's not a scalable strategy. And so, because it doesn't generate scalable evidence or regulatory learning, and doesn't necessarily create a repeatable access for the next patient. And what we really need to do is create that infrastructure. And that's what this master protocol is designed to do. It's designed to enable the development of a sustainable, scalable infrastructure. And if we're serious about individualized medicines and making it available to everybody, we need infrastructure. So I think that we can all agree that the real question is not, can we make individualized ASOs? We already know that we can. We've done it. We've done it over and over again. The questions really are, can we make an individualized medicine legible to regulators? And we're making inroads there based on the news today, as well as where the MHRA is going. And hopefully other regulators will start to turn their heads in that direction as well. We can we design a development model that produces structured data? And can we build a framework that doesn't collapse under the weight of heterogeneity? And also, can we create a system where the second child does not start from zero? I think both the MHRA as well as the FDA have signaled, as we heard today, openness to pathway innovation for highly individualized therapies. And importantly, the regulatory conversation is beginning to shift from reviewing every product independently to evaluating and overseeing a standard development process. And so this shift is really lowering an important barrier to scaling. And we believe that this enables something new at this moment. And this is what we're calling process approval. And here's what we mean by that. We are describing a regulatory recognition that under clearly defined scientific and manufacturing constraints, a validated development process can generate predictable quality and a justified expectation of safety and clinical benefit, enabling the systematic or authorization and deployment of individualized therapies that are the approved, covered, and reimbursable standard of care. And then this raises another question for us is, and this is, I think, the most important question is can we build a validated, regulator legible process that makes individualized medicines rather reproducible, audible, and also importantly, scalable? So to answer these important questions, we're deliberately building an ecosystem of academic, patient advocacy, regulatory, payer, and industry partnerships. And our partnership with GOSH and trusted regulatory relationships that we built through frequent interactions with organizations like the Rare Therapies Launchpad is central to validating individualized therapies under this master protocol framework. And together, we are validating this protocol that demonstrates individualized medicines can be systematically and also be produced, and data can be reproduced in a repeatable and regulatory manner. And this collaboration actually ensures innovation, clinical rigor and scalability that we are moving toward together. So with that, I'll hand it over to Paul to take us through the protocol. Paul?
Thank you very much, Michelle. So next slide, please. So we're talking today about the protocol, the master protocol investigation of individualized antisense oligonucleotides in people with unique genetic variants causing severely debilitating, life-threatening CNS conditions. Next slide, please. Michelle, in my clinic, I meet children with devastating neurodegenerative conditions. And these families bring with them experiences of years going through unbearable diagnostic odyssey, as some of you know. And when they finally receive a diagnosis, they hope that there will be treatment that could help them. But then we have to tell them something painful. There is no approved therapy, and they may never be because the condition is too rare. You talked about the ultra rare disorders. However, for some of these patients, science gives hope that potential therapies could be developed for specific mutations, and this creates a unique ethical pressure. This pressure on many of us. We as clinicians want to act, families want to act, but acting without structure, as you talked about, creates risk. It creates inconsistency. That's what compassionate use programs provide. And it makes it nearly impossible to learn systematically. What excites me about this master protocol is that it enables disciplined speed. It creates framework where treatment can occur under regulatory oversight with standardized assessments and with responsibility not just for one patient, but this responsibility is to future patients. Next slide, please. So we have developed a master protocol in collaboration with EOM, our team at GOSH and Institute of Child Health, and Tim Yu, who of course is here, that focuses on the evaluation of these individualized ASOs in people with unique genetic variants, causing these debilitating, life-threatening CNS conditions. I'm pleased to say that the protocol is part of a clinical trial application that was approved in the UK now by the drug regulator MHRA, and we have successfully enrolled and dosed our first patient. So this master protocol, EOM MP1, phase 1-2, multi-center, open-labeled, within participant dose escalation trial, investigating individualized antisense oligos in people with these conditions. These are unique genetic variants, and this is a prospective clinical framework that is designed to enable standardized data collection and pre-specified analyses across individualized ASOs for patients with these unique variants. By incorporating structured run-in periods within participant comparisons and harmonized safety, biomarker, functional, and quality of life assessments, the protocol is designed to generate a robust evidence base to inform regulatory decision-making and support the advancement of these individualized medicines at scale, while preserving the flexibility required for N of 1 therapies. So what we're not saying is that one gene, one mutation, one drug. Instead, we are saying something different. This is not a trial of single therapy. This is a trial of a process. Next slide, Michelle, please. This proof of concept protocol is designed as a basket trial, evaluating the process of ASO development rather than the product. Included in the protocol are assessments such as those for safety, pharmacokinetics, and various real-world assessments that will enable evaluation across indications, while also including indication-specific assessments that will contribute to the data supporting the evidence of effectiveness, not only for the individualized ASO, but for the overall process. Next slide, please. A new unit of assessment, platform process plus standardized chemistry rather than sequence by sequence reinvention. This is the core innovation. The sequence is individualized, but the backbone chemistry is standardized. And that means we can begin to treat individualized ASOs not as totally unrelated molecules, but as instances of a platform process that can accumulate learning. This is how we move from one-off rescue to scalable medicine. Next slide, please. ASOs are uniquely suited for that because class effects are driven heavily by chemistry. And when the backbone chemistry is consistent, we have something rare in individualized medicine, which is comparability. We can start to build an evidence base across different sequences because chemistry drives predictable pharmacokinetics and safety patterns. And we can monitor and manage known ASO class effects. This is not a leap into unknown. This is a structured attempt to turn what we already know into a scalable development model. Next slide, please. So here's the EOM MP1 protocol structure. Master protocol is agnostic to indication. It includes standard assessments applied to all indications such as safety, PK, mobility, and patient-reported outcomes. It also includes the meetings of the Independent Data Monitoring Committee and the dose escalation progress. Then there are indication-specific information, background, specific ASO development information, and specific assessments. Information and consent form ICF is generic with some specific appendices that explain indication-specific assessments. Next slide, please. We will enroll 10 participants in total, each with a unique genetic variant, a severely debilitating, life-threatening CNS condition, and no meaningful effective therapy. The first treated participant must be pediatric, and that means between 1 and 17 years of age at first dose. And that's intentional because this is where the unmet need is most acute and where disease progression is often fastest. Next slide, please. Safety is the primary endpoint. We assess dose-related incidence and severity of adverse events, serious adverse events, and adverse events of special interest. We'll monitor plasma and CSF laboratory exams, neurological exams, electrocardiogram, brain MRI scans. The secondary objective is pharmacokinetics. This data is critical to inform our dose escalation progress. This is a clinical trial designed for rigor. Next slide, Michelle. This protocol is unique in that it will systematically collect data to support a process approval. Prior to the development of this master protocol, most protocols for individualized therapies are bespoke clinical programs, largely with retrospective data analysis and ad hoc data sets. We are collecting an industry-grade data set where all assessments are pre-specified and systematically collected. All participants will undergo the same assessments to enable comparison across indications while also undergoing indication-specific assessments, contributing to evidence of effectiveness for that particular indication. During the pre-treatment phase in Part A, we will develop a baseline patient dossier of lab studies, biomarker, clinical assessments, and patient-reported outcomes, and perform an in-depth chart review to establish the disease trajectory as evaluated with multiple assessments, and then compare this data with that collected in Part B, the treatment period. During the treatment period, all patients receive 5 intrathecal doses of their individualized ASO. An IDMC reviews all safety and PK data prior to the escalation to the next dose. In a typical rare disease trial, you try to find a control group. Here, that may be impossible. When a mutation is unique, you cannot recruit a cohort. So the design uses within participant comparisons. We collect retrospective and prospective run-in data to define baseline disease progression, then compare on-treatment response to that baseline. This is how we build interpretable evidence in the absence of traditional trial structure. Next slide. So here just shows you again the trial design where there is a part A phase with a screening run-in where we collect as well as retrospective, also prospective data. And this is a minimum of 4 weeks, but could go as long as 12 to 15 months. And then there is a part B, which is the treatment period, and then there is an extension period. This cannot begin until the regulatory approval of ASO-specific investigator brochure and all the documentation that has to be supplied to the MHRA for each ASO. Then there is an administration of 5 ASO doses, and these happen no less than 4 weeks apart with the IDMC review after each of these administrations. There can be up to 3 participants with the same unique genetic variant treated with the same ASO. Next slide, please. So this details the assessments on the protocol, which are, as I said already, the safety assessments, PK, real-world mobility assessments, using some of it will be used using wearable technologies, and then the ability to perform various tasks that are more specific. Some of them are generic, and some of them are more specific. And then the laboratory analysis collected blood, CSF, and also the pharmacodynamic assessments that will be specific to different indications. Next slide, please. So the starting dose for all individualized ASOs is based on repeat dose rat toxicology studies using the intended clinical intrathecal route of administration and published literature of ASOs with similar backbone chemistry. The initial dose of individualized ASOs is justified based on the expected to have pharmacological effects and be reasonably safe. The selected starting dose includes a conservative margin of safety, and the pharmacological class of the study drug, an ASO, is associated with sequence-specific activity and generally favorable non-clinical safety profile, further supporting the chosen approach. Dose escalation is individualized, and escalation is not automatic but is dependent on the IDMC approval. This strategy reflects a risk-averse approach that controlled, deliberate, and ethically governed. Next slide, please. We also capture exploratory endpoints, and this is where the protocol becomes a data engine. We'll look at biomarkers in CSF and blood, look at the real-world mobility, video-based assessments, quality of life measures, collect qualitative interviews, and also cognitive and behavioral assessments and indication-specific measures. These measures are not nice to have. They are the foundation of regulatory learning, and they are the foundation of future access. Next slide, please, Michelle. So again, this is the safety monitoring that is going to happen, and there is a process level oversight with defined failure rate thresholds, pre-specified hold criteria, IDMC evaluation. And also there is a cross-molecule signal detection using pooled adverse event review, statistical boundaries and alerts, PK drift across ASOs, and also molecule-specific monitoring, continuous adverse events, and some of them halting. Some of the doses could be, we could stop moving to the next dose. Root cause analysis and IDMC evaluation happens in these cases. Next slide, please. So in fact, beforehand back to Michelle, I just want to mention that the teamwork with EOM has been excellent so far. There is willingness to help discuss rationally any current issues. Our GOSH team has a lot of experience running clinical trials in rare pediatric disorders, ultra-rare disorders. A lot of them are industry collaboration, and it has been one of the outstanding experiences working with EOM so far. There is a lot of support that I'm getting from the clinicians who are not directly involved in the trial, but working at GOSH, because they understand the importance of this work that can help the current patient, but also those patients that will need this treatment and can access this treatment in the future. As the hospital look after one of the largest group of rare disease patients in the world, we understand our own responsibility. Now back to you, Michelle.
Thank you, Paul. So I think that we can all see that this is more than a protocol. As we've seen there, it's really a shift in regulatory thinking. And I'm just going to review a few things here is that we know that Paul talked about and that I had talked about previously is that we know that individualized therapies cannot scale under a product by product approval model. If every molecule is treated as entirely new, access will just be limited. Our objective here with this protocol is process approval, and that means having regulatory confidence in a validated, reproducible developmental framework. If the process is standardized, then we have the infrastructure to ensure quality, safety oversight, dosing, and this systematic collection then leads to individualized therapies being available at scale. The master protocol, as Paul had said, was designed to evaluate the process and to provide a structured, high-quality data set evaluating clinical effectiveness as well, and not just a molecule. And we started in ASOs in children with severe single-gene neurodegenerative diseases in the UK because this is where there is the highest unmet need, the most clear biology, and also, I think, feasible regulatory engagement. So this is not theoretical. We've treated our first patient at GOSH. We're advancing additional ASOs into the same framework. And this repetition is really intentional because it builds robustness. So the goal is simple here is really to move individualized medicines from exception to standard of care. And just going into where we are today, having the protocol, having the availability of the FDA plausible mechanism guidance, we've compared sort of the protocol with the guidance, and you can see where the protocol lines up with the qualifications and the guidance domains that are required in the current document. So I just want to end with just a few questions because I think that as a community, we have to think about these things as we're moving forward. One of the things that we need to think about are, can regulators accept aggregated process learning? How do we interpret and detect signals amongst heterogeneity? These data sets are going to be heterogeneous. And perhaps I think most importantly, what does success actually look like? Because success is not just one miracle case report, it's a reproducible process. And just some thoughts on what success could look like from my point of view is that success is not just a dramatic responder story, and it's not when the next child or few children have a responder story. It's when these next children do not start from zero. It's when we have a standardized baseline data, harmonized endpoints, regulatory grade safety monitoring, as well as pre-specified analyses, and an infrastructure that really supports a repeatable process, allowing access to individualized therapies for everyone. And this is what our protocol is designed to help create. It's designed to generate that evidence base needed to support individualized medicines at scale. And so I just want to end with this, is that a world where the diagnosis leads to treatment should not depend on how common your genetic variant is. Precision medicine is not precision if only one child ever gets it. And the great thing is that we are not limited any longer by the science, and regulatory barriers, as we've seen today, are coming down. And so we're really together as a community looking forward to building the infrastructure to make individualized therapies accessible for everyone. And with that, I'd like to thank our brave and strong study participants as well as their families. Dr. Tim Yu for all of his support and guidance, Paul and his team for as well as being super collaborators and really being champions of this protocol at GOSH, and for recruiting these patients, as well as Haiyan Zhu and her lab, who has been really helpful in finding different mutations for us to consider. And also, obviously, the N1C Julia's organization, Mila's Miracle Foundation, as well as the support from the MHRA and the National Health Service. And we're looking forward to engaging with the FDA in the near future. So thank you.
Thank you, Michelle. Michelle, Paul, that was a really fantastically clear presentation. I just want to say that you guys are actually carrying forward the promise of this, and that means so much to all of us who are looking to make these treatments a reality for kids everywhere. So for those of you, I'm going to actually turn off my background. We're now this is an open Q&A right now. All I can start us off and I'll say like two things. One is that truly today we were actually in the the I guess the great hall of HHS when the plausible mechanism pathway was announced. So Julie and Tim are both here, and as you saw, for those of you who joined late, this has been the Mila to Millions crusade over the past
10 years.
now 10 years. And so this is how progress happens, and it really stems from people. And so we are so grateful for you guys as part of this community. If you think of the N1C as this society of interventional genetics, right? The professionals who are gathered here. It doesn't matter your identity, whether you are a patient, a family member, a professional drug developer, an academic. You all can be a part of this, and we encourage you to be part of this too. So I see some questions that are being dropped in the chat. What I will do is actually say, for now, I want to ask one juicy question is now, how does this get paid for, right? We talk about the reimbursement, and what do you guys think about that next step of if we have efficacy on an N of 1, if these things are all moving the way that you want, what happens to reimbursement models? So Michelle, would you be all right to take a crack at that?
Sure. Yeah, and this is something that we think about a lot, Winston, is how are we going to get these therapies reimbursed? And I think part of it comes from having that regulatory framework for approval. And once these therapies are approved, we then also have to engage with those payers and show them that we are building the database to support reimbursement. And part of that is looking at across these ASOs or whatever modality you want to put in that blank space. It could be ASOs, siRNAs, gene editing, and showing with that data that's been systematically and prospectively collected that it's safe, as well as also demonstrating what is clinically effective. And I think that as we move forward as a community, that we need to think about what is that package for clinical effectiveness look like? Because we're not doing these studies to look at population endpoints. We really need to think about personalized endpoints and what it means for that patient. Regulators really care about how patients feel, function, and survive. And I always say that individualized therapies need personalized endpoints. And so really trying to understand from the get-go what people want from their therapy and then prospectively evaluating what is meaningful to them. And that may not necessarily mean what those validated endpoints are. It may mean that a kiddo can feed themselves. Julia often says that one of the main benefits of the therapy from Millicent was the fact that Mila could actually sit up and play with her brother. And these things are meaningful to the families and the families that we're serving. And I think we as a community need to be thoughtful about proposing what that package looks like, and that if we are able to achieve those endpoints, that is something that is reimbursable. And frankly, that's the way we practice medicine, right? I mean, as a physician, if somebody is getting benefit from a therapy, I'm going to continue giving them that therapy. If they're not benefiting from that therapy, I'm going to take them off that therapy. I'm not going to say, well, the study says it should work, so it should work. We really base those decisions on individual therapy. So I think that this is about also educating payers about what the expectations can be. And that was one of the questions that I had asked during this presentation is like, what expectations do we want to set for these individualized therapies?
Yeah, and I'll chime in. I feel like one of the big milestones for today, at least in the FDA, is that this was a pathway towards approval and not just an IND. And
Right.
approval at least unlocks the pathway towards reimbursement. And there's definitely education ahead for all of us to do in terms of saying why it's worthwhile, how do we equate it to other therapies in which there may not always be the expectation of, or there's the expectation of benefit, but you may not always get the benefit, yet they're still covered, like surgical procedures and such. And that is a powerful analogy. I will now go through the some of the questions that came in. So Mark asked, how many ASOs will be included for your first trial? Yeah, and the second part is for those ASOs, do they have the same potencies and tox profiles?
So we're including up to 10 participants in the clinical trial, which means we could have up to 10 ASOs in the clinical trial. As Paul had stated, up to 3 people can be treated with the same individualized ASO. So it really depends on how many individuals we identify who qualify for treatment with that ASO. In terms of the design of our ASOs and the potency, when we design the ASOs, we keep the backbone stable. That backbone is a mixmer, and then we individualize the sequence. And they undergo a series of assessments that we use to make sure that we are optimizing the potency as well as reducing the toxicity for that particular ASO. I think that in terms of the backbone, all of us have done these experiments like hundreds of times. We know that for many of these backbones, you do the same thing over and over again, and you're showing the same safety. And then we can also reduce the potential for toxicity by doing assays in vitro. So we're really optimizing for decreased toxicity as well as increased predictability for clinical effectiveness with our screening processes for these ASOs.
Great, thanks so much. You answered a bunch of those questions there, and I'll also say that the I feel like one of the features of this protocol is that you don't have to have a pre-baked ASO in hand and just say we have these 10 ASO products. That there is a part of who is presenting in need, right? And then they would be there's the development process can occur under this existing protocol. Am I getting that correct?
That's correct. So we can enroll participants as we design as well as manufacture their ASOs, and that's a critical time period where Paul can enroll that patient into our protocol, and he can start collecting that data to develop that patient dossier and understand the baseline and their disease trajectory, but not just based on sort of anecdotal or retrospective data, but prospectively collecting that data with pre-specified assessments during that time period.
Absolutely. Wow. So this is a really great, what we'll do is actually take a download of the questions for this. And then, as I mentioned in the beginning, we have a forum called Connections that the N1C hosts for, actually we'll upload these questions there and get that conversation going, offline. So in case we run out of time here. So the conversations with the FDA or EMA, I believe this is actually in the UK, so it's the MHRA. Scott, you were asking about, is the trial approved for accrual? You mean enrollment, right? So I believe that, yep, go ahead.
No, exactly. Forgive me, I was 10 minutes late. And so I just didn't quite understand. I saw all the stuff that you were, first of all, bravo. I don't know. I mean, just magnificent. So thank you so much for presenting this. But I saw the attempt to harmonize what the FDA wants, but has there already been pre-IND discussions and all this is kosher, so to speak, with what they want, or through an actual conversation, or is it still at this stage where that'll be a step down the line?
Yeah. So Scott, the protocol, as we said, is approved with the MHRA, and we've
It
already
is.
enrolled and dosed our first patient under the
Well,
protocol in the MHRA. And this protocol is the first protocol that the MHRA approved for a master protocol for multiple indications to be treated by different ASOs. So and they were fantastic. We engaged with them very frequently and incorporated all of their feedback, and they were very helpful, along with guidance from Paul and his team in developing this protocol. In terms of the FDA, this guidance is promising, and at the end, I sort of did a quick, side by side comparison about what they're saying in the guidance, and the protocol does seem to line up with what the guidance is suggesting, and so we will be engaging with them at some point in the future.
Great.
Thank you so
The
much.
the next question is about just will these documents be made available? And I believe what the goal of the N1 collaboratives is how enable the sharing. And actually, EOM has very generously provided some of these documents that will be redacted just to protect some proprietary information as well as patient-specific information there. But the structure of it is supposed to really inform the entire community and moving forward these life-changing treatments. All right, going down the list here, access to these trials. Paul, maybe this is something that you can speak to of just enrollment as well as how does one get in touch and become eligible for these trials?
Yeah, sure. Thanks, Winston. Well, this is an industry-sponsored trial, and it is fairly standard how the access happens. The difference here is that when the patient first contacts us, they would not have the actual drug. There wouldn't be an ASO ready. So the initial contact could go through the trial team, or it could go through the company. But essentially, the ASO would be the actual case would then be discussed by a committee that is, essentially an EOM committee that has representation from various clinicians, scientists, and they would discuss the essentially how likely it is that the ASO can be developed for that particular patient. Does the patient fit the criteria of severely debilitating CNS condition? And whether the ASO would actually have a meaningful effect in that particular patient. So the simple answer to the question how to contact, I think the contact can be through the clinical team at GOSH that are involved in the trial, or can be there would be I think Michelle could explain how they could contact directly through EOM. But that's not essentially the difference in the process is that then the committee will look at that.
Yeah, so that's right, Paul. And I think other ways that patients can become involved is that they can go to the website, everyonemedicines.com, and there's a link there that says contact us. And that will take you to a platform where you can provide your information and we can start the process of potential evaluation.
I guess the other thing to add is when people are from all over the world and how could they access? So at the moment, the trial is only open at GOSH. So, and only 10 patients the aim is to recruit 10 patients. So it is possible that patients from abroad may be able to access this. But of course, there is only a limited number of patients, and I don't know what EOM are planning to do after they've recruited 10 patients. Because of course, that's a small number.
All right. So, Amy, we will get back to your other part of your questions, which is a really critical piece about access and making sure that it's equitable. I would say making sure that there's a pathway towards reimbursement that is key so that, families are not holding bake sales and golf tournaments to try and raise money to make these programs happen. So that's maybe my perspective, but we'll get back to that. Eric had a question about And thank you for a biostatistician joining us because there is an incredible amount of work with Bayesian stats and other things that we could use your help on to provide some statistical oomph to these N of 1 trials. You mentioned for insights from HCA experts on feasibility and appetite. Do you mean of the trial itself, or do you mean like, yeah, can you explain more about which? Are you talking about like what the evidentiary standard they expect to see, or tell us more what would be helpful?
Winston, I think that because these are individualized therapies and the data that we are generating, we want to have a data set that supports a process. I think that there needs to be an expectation setting of what you are going to actually see as a result of these data sets. Obviously, we're never going to hit statistical significance. I mean, there's just too few patients. And even if there are a few patients, you're not going to hit statistical significance. And so, what we need to think about is what is the data package that's needed to show clinical effectiveness for these patients in order to support continued treatment for them, not in order to support, I guess, not only for the process but also for continued treatment, for payers and things like that, and setting that example and being upfront about it and having set standards. And I think that we're at that point where the community itself can help set those standards, and whether that be by inferential statistics. Obviously, frequentist statistics are a bit more difficult because of the patient numbers, but using Bayesian technologies or Bayesian methodologies, or inferential statistics, we can certainly look for patterns in terms of clinical effectiveness. And so this is where actually a lot of our thinking is right now, is trying to look at different data sets to understand how do we make sense of these heterogeneous data sets. So not only take the data from part A, which all patients undergo those assessments. So those assessments are safety assessments, the PK, making sure that the PK behaves the same, real world assessments, as well as PROs. That's pretty standard across, in that you can actually look across indications. But where things can potentially be a little bit messier is when you get into the indication specific assessments and really trying to decide, okay, like how do we take these heterogeneous data sets where you're looking at different biomarkers or different assessments, or using EEGs or MRIs in a different way, and how do you take these heterogeneous data sets to say, okay, these are the assessments that we're going to do that are going to support clinical effectiveness? Is it that we see improvement in one of these assessments, 2, 4? Do we break them up into categories, or do we weight them? And so these are things that I think that we as a community need to think about in terms of setting that bar for what we mean by clinical effectiveness for individualized therapies. Paul, do you have any thoughts on
Michelle.
that as well?
Well, it's a difficult question, isn't it? And I think that's you are paving the new path for platform approval, platform technology approval. I was actually at a meeting with MHRA about a different platform. So I think the regulators are becoming, as you've said, alluded to the FDA as well, open to these platform approvals. How do you put together unrelated conditions, unrelated ASOs, so there is something that they have in common, that's certainly safety. It is difficult, but we'll see how it works. So I think it's to be developed still. Reimbursement, of course, is a key question, Winston, that we still need to solve. And I think also for different countries, the reimbursement question is different. With the FDA, is once the ASO is licensed, it will probably be reimbursed. In the UK, it's very different. And in the UK, there is no such process where you can assess these individualized medicines. So that needs to be developed still. Although the regulators seem to be walking towards that platform approvals, which is a good thing for us.
We have 5 minutes left, and I'm going to try to bundle some of these questions together. So, Mathurin, you mentioned about the technical process itself as well as the cost. The I guess that this is something where at a high level, could either Paul or Michelle comment on just the process of designing these, um, what's the funnel of going from, different leads to the actual development candidate and what goes into a patient.
So once a patient is identified, as Paul had said, we look at the mutation, and then we meet with a committee to decide whether that mutation is causative of that patient's symptomatology, as well as to evaluate whether the mutation or genetic variant that has been identified is ASO amenable. So meaning that there is a way to develop an ASO for that specific gene variant. Once we have this meeting and have a discussion about about that, and we also discuss a potential approach. And then we work through doing target identification and intelligence, as well as using different AI-enabled platforms to help us really identify those ASOs that are the most likely to provide high efficacy, but the least toxicity. And we do have requirements for that design. I don't know the exact requirements because I'm not a bioinformatician, and so I can't talk specifically to them, but related to the design, we're looking for favorable characteristics of each ASO that could be potentially designed. And because our backbone is a mixmer and it's stable, we already know the adverse event profile, and we're able to reduce the potential toxicity from the sequence, the individualized sequence, by looking at specific assays. And these are typical assays that are used, I think, in probably everybody's lab. And so by using that process, we're able to whittle this down from, several hundred to a few that we do take for assays, as well as functional assessments. And then from those tox assays and functional assessments, then we nominate that ASO.
Great. Thank you so much for that detailed response there. Mathis and Rebecca had a kind of related questions where one of them was describing, the safety as a primary outcome, and is that together is that alone able to achieve platform approval without efficacy? And maybe I'll just jump in a little bit and say, to me, this seems like a really important first step, right? That's we need efficacy to show this, but there needs to be. It seems like the innovation here is really that there's no structure that's been enabling of this before. But I'll pass it to Paul, who you may have had conversations with MHRA and then NHS afterwards.
Yeah, I think I completely agree that safety is important, but I also agree with Mattis that it is important to have some efficacy. But the hope is that the threshold will be lowered for these individualized therapies, because as we said before, we can't have a controlled group. It will be an intrapatient controlled. It's sort of statistical significance would be very difficult to demonstrate. So what we hope is that biological plausibility that you can show in vitro and then show some evidence based on biomarkers, based on patient-reported outcomes, will build a picture. For example, MHRA today talked about specific cases, not perhaps just a sort of a black and white, yes, filling the forms, but actually parents talking and saying, this was what it was like before, this is what it's like now. So these stories actually will help and will have a significant weight. That's what MHRA say now. So I think the hope is that the threshold can be lowered, the approach will be different, and it's not so much the standard statistical methods.
Thank you, Paul. Thank you, Michelle. I just think that that's what a wonderful treat to hear from two deep experts in the development and realization of our goal of going from Milo to millions, and on a very special day. We hope you will not only continue the conversation on connections, but join the N1C. Like if you have a protocol or things that you are excited about sharing, if you have statistical things that could be useful to our community, this is the whole point of why we're bringing us together so that we can grow as a community and advance this field. So thank you all. This has really been a wonderful turnout. And yeah, thank you, Julia and Tim, for the amazing cameo here.
Thank you very
Thank
much.
you, everybody.
Bye-bye.
Thanks, you
Congratulations, Paul and
guys.
Michelle.
Thank
Thank you.
you.